Hi everyone, my name is Genna. I'm here at the Northwestern University Lab, and today I'm going to be presenting my project on 2D phosphoproteome mapping of spleen tissue. So the spleen is located in the upper left abdominal cavity and is responsible for filtering the blood. There are two main sections of the spleen, the red pulp and the white pulp. So as you can see here, the white pulp is spotted throughout the red pulp, and the red pulp is surrounding. The white pulp is known as the germinal center and has an accumulation of lymphocytes and other immune cells around the arterial vessel. And the red pulp here consists of vasculature, which carries blood in and out of the spleen and is made up of other lymphatic cells like macrophages. And so the overall goal of my project is to create a visual representation of protein arrangement in spleen tissue through bottom-up proteomics and analyzing protein expression data by identifying potential gene markers and the pathways associated with the red and white pulp regions of the spleen. So here's the streamlined workflow created for this technique of proteomics. First, there was a sample collection done using the laser capture microdissection tool, and the samples were cut at about 200 by 200 micrometers with about 10 micrometer thickness. And so here to the left is a picture of the spleen tissue before it was cut, and here to the right it shows the 12 by 20 grid that was created and where the voxels were cut from. Next, it was processed using the Surface Assistant one-pot processing, and this was used to lyse and digest the sample, and then it went through TMT Pro labeling. This was used to enhance protein detection and sample throughput and to ensure that we did not lose protein detection. Next, the sample was loaded into the tandem C18 IMAX C18 column for phosphoproteome analysis, where the phosphopeptides in the column were used for the phosphoproteome, and the flow through was used for the global proteome analysis. And lastly was the LC tandem MS, which was used for protein and phosphoprotein detection and data collection. So going on to our results, we were able to identify over 900 phosphopeptides in each TMT set, with about 600 being quantified, and we were able to identify over 2,000 proteins, with just about the same amount being quantified. And overall, there was more data collected for our global proteome data set than that of our phosphoproteome data set. The plots here in the middle represent the principal component analysis. So in these plots, the red dots indicate the white pulp regions, whereas the blue dots indicate the red pulp regions, and the green dots represent an unknown region. And so using the quantified protein and phosphopeptide data, we looked for gene markers that could help distinguish the red and white pulp regions here. And so we created several 2D heat maps using R to visually represent the expression levels of the proteins and the peptides across the tissue. So first we looked at several different white pulp gene markers using the global proteome data. So here CD45 is the leukocyte common antigen, and is expressed on all hemopoietic cells and is essential for the activation of T cells. And CD47 here is an integral transmembrane molecule, and it plays a role in MCH class 2 molecules and is expressed in B cells and B lymphocytes, which are present in the white pulp. So looking at the expression data here to the right, and this is a heat map, and the blue voxels here show a relatively higher expression, whereas the red and orange voxels show a relatively lower expression. So the red pulp here is indicated with no markings, and so we're calling that cluster 1. The white pulp here is shown with a crosshatch pattern, and we're calling that cluster 2. And then over here is cluster 3, which is the unknown region. And for our purposes, since cluster 3 was had such a low expression level of all the genes, we decided to remove that from our data to get a better look at the differentiation between the red pulp and the white pulp. And so looking at the expression values for CD45 and CD47 here on the side, you can see that there's overall a more blue blue-green voxels in the white pulp region versus the red-orange voxels that are in the red pulp region. Next, we identified red pulp gene markers. And so here, CD36 is a transmembrane glycoprotein receptor, and is expressed on various cells, including monocytes, macrophages, and can be found in the red pulp of the spleen. Similarly, CD163 is expressed on monocytes, macrophages, and specifically red pulp macrophages. So we know that this is going to be highly expressed in the red pulp. And here to the right, looking at the expression value maps of both CD36 and CD163, you can see that there are more red-orange voxels in the white pulp region versus the blue-green in the red pulp region, and the same for CD163, showing that there is an overall lower relative expression level in the white pulp versus the red pulp. And next, we looked at identifying different phosphopeptide markers using the phosphoproteome data. So LSP1 is a gene that encodes for intracellular F-actin binding protein, and this is expressed in lymphocytes, neutrophils, and macrophages. And then we looked at IL-16, which encodes for interleukin-16 and is found in lymphocytes, monocytes, and many other immune cells, and is known to induce T-cell lymphocyte expression. And so looking at the heat maps here to the right, we can tell that there's definite higher level of phosphorylation in the white pulp regions because of this the green-blue voxels here versus the red and orange voxels in the red pulp region. And the same for IL-16, there's a higher phosphorylation level in the white pulp versus the red pulp. So next we did a pathway analysis. So to do this, we found the z-score of each voxel using the collected expression values for each gene. And then we summed the z-score for the upregulated proteins. And we summed the expression for the upregulated proteins and we mapped it using R. Here we're looking at the erythrocyte pathway, and this is used to uptake carbon dioxide and oxygen in the body, and vice versa. And so the spleen holds on to red blood cells and is used to filter the erythrocytes by their physical characteristics. So there's going to be an upregulation of erythrocytes and the erythrocyte pathway in the spleen. And so here we used CYB5R1, HBB, HB1, SLC4A1, CA1, and CA2 to do this analysis. And looking at the heat map here, you can see that there is an overall lower expression in the white pulp, just looking at the red and orange voxels versus the red pulp with the green and blue voxels. And looking here to the right, this is a volcano plot of showing the overall expressions of the data set. And so you can see that there is an upregulation of the genes used for this pathway in terms of the overall data set. Next, we looked at the T-cell signaling pathway, and this pathway plays a key role in functioning of T-cells by providing a connection between the T-cells and the antigen-presenting cell. And so the genes used in this one were PLCG2, HLA genes, and TFAM. And so looking at the in the middle plot here, you can see that there is an overall upregulation of these genes in the white pulp versus the downregulation in the red pulp by looking at the red and orange colors. And then looking at the volcano plot here to the right, you can see that there is an overall downregulation of these genes in the overall data set, and that's because this is normalized based on the expression of red pulp over white pulp, meaning that there's a lower log2 fold change. And lastly, we looked at the heme signaling pathway, and so since heme is attached to the hemoglobin and is present in many of the red blood cells, when the spleen is filtering old red blood cells, the heme signaling pathway is present and is a good marker for the red pulp. And so looking here at the middle plot, you can see that there is higher regulation of the heme signaling pathway in the red pulp versus the white pulp, which has the orange and red voxels, and the genes used for this were HPA1, HBB, and HMOX1. You can see here over to over to the right, that there was an overall upregulation of all these genes in the overall data set. And finally, my conclusion in this analysis, we were overall able to characterize the red and white pulp regions of the spleen tissue by identifying key gene markers and analyzing the different cellular pathways. And so we use this in order to create a more comprehensive analysis. We can use different methods to have a more robust analysis. We can investigate the expression levels of cluster 3 and the potential markers of this region. We can utilize a higher resolution to map proteins on the single cell level, and we can integrate other proteomic platforms like Codex for differential protein expression validation. And finally, here are just some acknowledgements. Thank you to the HuBMAP Consortium and the Lube Lab at Northwestern for enabling me to partake in this internship. Thank you to the members of the Lube Lab, our collaborators at PNL and the University of Florida, and a special thanks to Ajay, Zarina, Maya, and Julie for having a smooth and productive internship. Thank you.